Picture a train sitting at a station. Passengers have boarded, conductors have checked tickets, and everything appears ready to go. But if the engineer's watch has stopped, the train never departs. The doors stay open, the whistle never blows, and the journey never begins. That's essentially what happens inside a cell when its developmental clock breaks down. An organism may never progress through the stages needed to reach adulthood, not because it lacks the parts, but because nobody's checking the time. Scientists at Cold Spring Harbor Laboratory, CSHL, have now found the engine's timekeeper, a master developmental clock that controls biological growth from embryo to adult. The discovery, published in early June 2026, reveals how cells keep their schedules in lockstep and what happens when the clock fails. The researchers studied C. elegans, a tiny transparent nematode worm that has served as a model organism for decades. Scientists already knew that development in C. elegans is driven by pulses of gene expression, bursts of genetic activity that fire in sequence, guiding the organism through each growth stage. What no one knew was how those pulses were timed so precisely. Enter MIF1 and LIN42, two proteins that form a feedback circuit acting as the worm's central developmental clock. Together, they determine when each pulse begins and how long it lasts. According to the team, this is the first example of a non-repeating biological clock. Unlike the circadian clock that ticks in 24-hour loops, this one marches forward like a ratchet. Step 1, the trigger. MIF1 kicks off each developmental stage. It's the green light that starts the engine. Step 2, the regulator. Once MIF1 fires, it activates LIN42. LIN42 then controls the intensity and duration of the genetic pulse, like a volume knob and timer rolled into one. Step 3, the checkpoint. MIF1 is also required to mark the stage's completion. Both the start and finish lines are its responsibility. Step 4, the ratchet. The circuit runs in all cells simultaneously, and every independent cellular clock stays in sync, but it only goes one direction. Development advances, it doesn't rewind. When researchers blocked MIF1, the entire developmental program collapsed. "We've never seen anything like this before," said CSHL Professor Christopher Hamel. MIF1 is part of this master regulatory clock for all cells, but it's also acting as a key maker and the master key for each stage of growth. This isn't just worm biology. The MIF1 per LIN42 circuit represents a fundamental design principle that may apply across species, including humans. Developmental disorders. Many conditions that cause growth abnormalities may trace back to timing failures rather than missing parts. If the clock runs fast or slow, development hits walls or skips stages. Cancer biology. Cancer is, in part, a disease of out-of-control growth, cells dividing when they shouldn't. Understanding what keeps the growth clock on schedule could reveal new therapeutic targets. Regenerative medicine. If scientists can understand how to synchronize cellular clocks across tissues, they may gain new tools for tissue engineering and organ regeneration. Perhaps the most intriguing unanswered question is communication. If every cell has its own MIF1 per LIN42 clock running in sync, are those clocks talking to each other? "We've never thought deeply about that question before," Hamel admitted. The CSHL team, which includes Director of Research Lima Joshuator, is now using AlphaFold and advanced sequencing to map exactly how MIF1 and LIN42 physically interact, and whether cellular clocks send signals to one another during development. 1. Development needs timing, not just parts. Cells don't just grow, they grow in order. The MIF1 per LIN42 ratchet ensures the sequence never reverses. 2. It's a first-of-its-kind clock. Unlike circadian rhythms that repeat, this clock moves forward once. Biologists may need new vocabulary to describe it. 3. The implications stretch far beyond worms. The same timing principle likely exists in humans, with direct relevance to developmental medicine, cancer, and regenerative therapies.